Welcome to Biochem Insights with Dr. Shefali.
In this video, we are going to talk about DNA replication. And the significance of learning about DNA replication is that only when you know the basic steps of replication, you will be able to understand all the application aspects of DNA replication. So what are the application aspects of replication? It's nothing but cloning techniques. So if at all you want to understand about recombinant DNA technology and about polymerase chain reaction, you will have to know about the steps of replication. So I will walk you through the entire DNA replication step by step, and we will also explore different enzymes and proteins involved in the process along the way. So let's begin with what is DNA replication. DNA replication is a biological process of producing two identical replicas of DNA from one original DNA molecule. In simple words, we can say that DNA replication is a process through which a DNA molecule makes a copy of itself. Yeah, DNA replication is a process through which a DNA molecule makes a copy of itself. Now the question arises here is that why is it essential for DNA to be replicated? What is the purpose of DNA replication? So DNA replication is essential to maintain the genetic continuity in the newly formed cells during cell division. Let us understand what does this mean. So whenever a cell divides or undergo mitosis, the two identical daughter cells are formed that must contain the same genetic information or DNA as the parent cell. Therefore, in order for the cell to replicate or divide, the DNA within the parent cell should also replicate. Let's see how it happens. As you can see here, this cell has 23 maternal and 23 paternal chromosomes. So the DNA within this chromosome and the DNA within this chromosome has to replicate to give 2 of each. After which the cell undergoes mitosis and you get two identical daughter cells. So the whole purpose of DNA replication is to allow for cell replication or cell division. This brings to the next question, that is, in which stage of cell cycle does DNA replication takes place? So this is your cell cycle where the cell enters in G1 phase, then S, G2 goes into mitosis and produces two identical daughter cells. So S phase is where DNA is copied or replicated. A good way to remember this is that S stands for synthesis, which means to make, and so therefore DNA replication occurs in S phase of cell cycle. Now let's discuss the salient features of DNA replication. Number 1, DNA replication is semi-conservative. Number 2, replication occurs in 5' to 3' direction. Number 3, replication is semi-discontinuous. And number 4, replication is bidirectional. Now let's see why DNA replication is semi-conservative. So during DNA replication, each of the two strands that make up the double helix first separate and serve as a template from which new strands are copied. The new strands that are synthesized will be complementary to the parental strand. That is, adenine in the parental strand will always pair with thymine in the daughter strand, and cytosine is always paired with guanine and vice versa. So if you look at the two daughter DNAs, in both the daughter DNAs, only one of the two strands is new, the other one is old, which you have partially conserved, and that is why DNA replication is semi-conservative. So in DNA replication, first two strands of DNA separate. Both the strands act as template. Both strands synthesize two new strands of DNA. The new strand will be complementary to the old strand. Each new daughter DNA consists of one new strand and one old strand. And this is why DNA replication is semi-conservative process. The next really important thing that you need to know is that DNA replication occurs in 5' to 3' direction. Why is that so? Because the enzyme DNA polymerase, which is responsible for synthesizing new strand, synthesizes new strand only in 5' to 3' direction, and for this DNA polymerase requires a 3' 5' strand to act as a template to synthesize a new strand in 5' to 3' direction. DNA replication is semi-discontinuous. As you can see, one of the strands is synthesized continuously while the other strand discontinuously in the form of fragments. You will understand this better once we discuss the steps of replication. Next is replication is bidirectional. DNA replication occurs in both directions from the origin of replication towards the replication fork. Again, you will be able to understand this better after we discuss the elongation step of replication. For now, you just observe the diagram and notice that one strand which is continuous in one direction, the same strand is synthesized discontinuously in other direction. The reason for this you will be able to understand on learning the steps of replication later in the video. Now let's start by listing basic functional requirement for DNA replication. Number 1, enzyme DNA polymerase. DNA polymerase is one of the most important molecules in DNA replication, as DNA polymerases are enzymes that synthesize DNA by adding nucleotides one by one to the 3' end of the growing chain, making sure that these nucleotides are complementary to the template strand. So the second requirement for DNA replication is a template, which is nothing but 2 strands of parent DNA. We have already discussed this earlier that DNA polymerase synthesizes DNA in 5' to 3' direction, and therefore what should be the polarity of the template strand? The template strand must be anti-parallel, right? So the template has to have 3', 5' orientation for DNA polymerase to synthesize a new strand in 5', 3' direction, and the new strand will be complementary to the template strand. Next is primer. So it is important to know that no known DNA polymerase is able to initiate polynucleotide synthesis on its own. It is only capable of adding nucleotides to the growing chain, and therefore it needs a primer to start the process. And primer is nothing but a short stretch of RNA nucleotides. Number 4 is substrate. We must know that building a new strand of DNA will require an energy source, and therefore all DNA polymerases require all 4-deoxynucleotides triphosphate. Now what does this mean? This means that they need deoxy-ATP, deoxy-GTP, deoxy-CTP and deoxy-TTP. So you will have to supply all 4-deoxynucleotide triphosphates for replication to happen. Fifth requirement is that they need magnesium or manganese or any divalent cation to act as catalyst. So why do they need these divalent cations, as you know that any enzyme itself is a catalytic substance? So DNA polymerase needs a divalent cation to improve its catalytic rate. And then it needs a buffer to maintain the pH of chemical reaction. See, ultimately what are we doing with the help of DNA polymerases? We are meditating a chemical reaction, and you know for any chemical reaction pH needs to be maintained, so we need a buffer. Now along with DNA polymerases, which is the master enzyme that carries out the synthesis of DNA, there are a number of other enzymes and proteins that work together to ensure the accurate and efficient duplication of DNA. So seventh and the last requirement for DNA replication is enzymes and proteins in addition to DNA polymerase. For example, single-stranded binding proteins, helicase, topoisomerase, primase, ligase, etc. Since the names and functions of the enzymes and proteins can get confusing, we will make an enzyme protein chart before we go over the steps of replication. And as we discuss the steps of replication, we will fill in our enzyme protein chart to keep all our information organized. Let's start with the stages of replication. The process of replication is broadly described as having 3 stages: initiation, elongation, and termination. Let's begin by considering initiation stage. We know that DNA molecule is made up of 2 single DNA strands twisted around each other in a double helix shape, right? As you can see here, this is prokaryotic circular DNA and this is linear eukaryotic DNA. We also know that replication can occur only from single-stranded DNA template. Why? Because the enzyme DNA polymerase, which is responsible for DNA replication, uses only single-stranded DNA as template for DNA synthesis. So in order for the 2 complementary strands of the parental strand to be replicated, What is the first and the foremost requirement? Obviously, they must separate. So the first step of replication is complementary strand separation. Now these strands cannot separate just from anywhere. So we need to identify the site of initiation from where the two strands start to separate. To understand the process better, let's consider the step ladder model of DNA. So again, this is prokaryotic circular DNA and this is eukaryotic linear DNA. As I told you that the first step of replication is complementary strand separation, and the site of initiation from where the two strands start to separate is called the origin of replication or ORI. As you can see, prokaryotic DNA has only one ORI, whereas eukaryotic DNA has multiple origin of replication sites. Now, what is the origin of replication and what is so special about these regions to be the site for initiating the two DNA strands to separate? So it is a segment of your chromosome which is rich in AT sequences, which means there are particular nucleotides in this region which are highly concentrated in adenine and thymine. So what did I say? DNA strands start to separate at origin of replication, and origin of replication is an AT rich region. Now just think, why do we want to pick AT rich region and not GC? Because there are 3 hydrogen bonds between G and C, whereas only 2 hydrogen bonds between A and T, and therefore adenine and thymine requires less energy to break those hydrogen bonds as compared to guanine and cytosine, and thus AT rich region is called DNA unwinding element. Once origin of replication is identified, origin of replication binding proteins, also known as pre-replication protein complex, binds to both the strands. In prokaryotes, it is called DNA-A, whereas in eukaryotes, it is ORC. So, origin of replication binding protein binds to both the strand and initiate unwinding by separating adenine and thymine nitrogenous bases, and thus successively denatures the DNA locally and form a bubble called as replication bubble, which is nothing but a short localized region of two single-stranded DNA. So now let's add ORC to our protein enzyme list. So the function of ORC is that it denatures the DNA locally to form replication bubble. So this is the magnified view of the replication bubble. Now, because these two strands are complementary to each other, they have a tendency to reanneal. What is that? It means they try to come back together in their original twisted double helix form. So, another protein named single-stranded binding proteins binds to both the strands and perform two functions. First, they prevent the single strands from reannealing, and second, they protect them from nucleases. Now, how do the nucleases harm the strands? By trying to break the phosphodiester bonds of single-stranded DNA. So what do these single-stranded binding proteins do? They act as a barrier and protect the single strands from reannealing and from exonucleases and endonucleases. So adding single-stranded binding proteins to the list, and their function is that they protect from reannealing of separated strands and nucleases. In each replication bubble, there are two Y-shaped structures called replication fork, in which the tines of the fork represent the separated strands. It is due to the replication fork that the replication bubble grows in both the directions. Let's understand it this way. So once the replication bubble is formed, all the enzymes start acting from the center of the bubble. And from center of the bubble, one set of enzymes act in one direction and another set of enzymes act in the other direction. And that is why we say that replication is a bidirectional process. Now DNA synthesis takes place inside the bubble in semi-discontinuous manner, as you can see in the diagram. The reason for this we will discuss later in the video. So this is one half of the replication bubble, and as you can see, two single-stranded DNA are guarded by single-stranded binding proteins to protect them from reannealing and nucleases. And where do you find all the enzymes for now? All the enzymes for now are at the center. So from the center, an enzyme called primase, which is a RNA polymerase,
binds with another enzyme called helicase to form primosome. So the primase within the primosome complex catalyzes the synthesis of primer. If you remember, in the beginning we discussed the requirement for DNA replication, where I told you DNA replication needs primer, which is a short segment of single-stranded RNA. So the question here is that we are doing DNA replication, right? So why do we need RNA here? So the fact is that the enzyme DNA polymerase, which is responsible for DNA synthesis, cannot initiate synthesis of complementary strand on ' own. It is only able to add nucleotides to an already existing nucleotide chain. So we need a primer that has free 3' OH end to which DNA polymerase adds nucleotides to elongate the chain. And so RNA primer serve as a starting point for DNA polymerase to be able to carry out DNA replication. So what is a primer? So primer is a short stretch of RNA. It is complementary and anti-parallel to the template strand. So this is the template strand which has 3', 5' orientation from left to right. So the RNA primer will be anti-parallel and will have 5' to 3' orientation in the same direction, right? Also, it will be complementary to the template strand. So adenine in the template strand will have uracil in the RNA strand. Similarly, guanine will have cytosine and so on. You must also know that RNA primer has a free OH group at the 3' end. I have already told you in the previous slide that DNA polymerase adds nucleotides to this 3' OH end of RNA primer. So adding primase and primer to the list now. Primase synthesize RNA primer and primer serves as starting point for DNA synthesis. Now let's suppose from the center the bottom strand is in 3', 5' direction towards the replication fork. And again the top strand would be anti-parallel, so 5' end would be here and 3' end would be here. Now what is the first requirement of replication? I have been telling you repeatedly that DNA replication requires 3', 5' strand to act as a template. So from the center the enzyme primase identifies and reads bottom strand in 3', 5' direction, against which it synthesizes RNA primer in 5' to 3' direction. Now how about the other strand from the center? The other strand is in which direction? 5' to 3' direction. And against this 5', 3' strand, do you know any enzyme which can synthesize a new strand in 3', 5' direction? No. There is no enzyme on this earth that can synthesize a new strand, be it DNA strand or RNA strand, in 3', 5' direction. So for the top strand, the enzymes know that they have to act from the angle of separation. So from the center, primosome reaches the angle of separation. What would be the polarity of top strand now? The polarity of the top strand would be 3' to 5'. And so now primase of the primosome complex will read the top strand in 3', 5' direction, against which it synthesizes RNA primer in 5' to 3' direction. Now enzyme helicase, as you can see at the replication fork, also has a very important function to play in the replication process. So as and when required, the helicase moves into the neighboring double-stranded region. It breaks the hydrogen bonds between the strand, forcing the strands apart and unwind the DNA double helix. And this is very important for the replication process to proceed. Here I want you to remember that helicase requires tons of energy in order to perform this function. So from where can the helicase get this energy? Yes. So ATP provides energy to the helicase to unwind the double-stranded DNA. Now as the DNA is opened up, the DNA double helix ahead of the replication fork experience torsional stress that leads to supercoils called as positive supercoils. So the three facts you need to remember about helicase are: first, its function, that helicase unwinds the double-stranded DNA at the replication fork. Number two, it uses energy in the form of ATP. And third, it creates positive supercoils. So what do positive supercoils do? They stop the further movement of helicase. And what will happen if the movement of helicase is stopped? Replication will also stop. So before we know more about positive supercoils, let's first add enzyme helicase to the list of enzymes and proteins. So enzyme helicase and its function to unwind the double-stranded DNA. So what I told you about positive supercoils, that as the DNA is opened up, the DNA double helix ahead of the replication fork experience torsional stress that leads to supercoils called as positive supercoils. Now to solve the problem of supercoils, there is a group of special enzymes called topoisomerases for removing supercoils. Now how do they do it? How do topoisomerases remove supercoils? So topoisomerases show nuclease and ligase activities by which first they cut the coiled DNA strand and then rejoins the relaxed DNA strand so that the process of replication is continuous and uninterrupted. So now adding topoisomerases to the list, and its function is to relax the supercoils caused by the movement of helicase ahead of the replication fork. So this is all about initiation stage. Let's quickly recap what we have seen so far. So number one, DNA A or ORC denatures double-stranded DNA locally. Separation of two strands of parental DNA results in the formation of replication bubble and replication fork. Single-stranded binding proteins binds to the exposed single-stranded DNA, thus prevent the strands from re-annealing and degradation. Primase synthesizes short stretch of RNA sequences called primers. Primers serve as a starting point for DNA synthesis. Helicase moves the replication fork forward by unwinding the DNA helix, and enzyme topoisomerase solve the problem of supercoiling. So the next stage is elongation, where DNA polymerase 3 actually synthesizes DNA. So here we have one half of the replication bubble and replication fork. The two separated DNA strands acting as template, single-stranded binding proteins keeping the two strands apart, helicase and primase, and primer at 3' end of both the strand. Now considering the lower strand first, where enzyme DNA polymerase 3 comes into play and gets attached to the 3' OH of RNA primer. As I've been telling you right from the beginning, that DNA polymerase 3 is the master enzyme that actually performs DNA synthesis. So let's see a few facts about DNA polymerase 3 to understand how does it perform DNA synthesis. So first, DNA polymerase 3 is DNA dependent DNA polymerase. It exhibits 5' 3' polymerase activity. It exhibits 3' 5' exonuclease activity, and it synthesizes leading and lagging strand. So what is DNA dependent DNA polymerase? DNA dependent DNA polymerase means that DNA polymerase requires DNA template to catalyze the polymerization of deoxyribonucleotides. In other words, DNA polymerase 3 requires a DNA template against which it synthesizes a new strand, and hence it is called DNA dependent DNA polymerase. You must also remember that DNA polymerase catalyzes the synthesis of DNA strand complementary to the given DNA template. Next is 5' 3' polymerase activity. To understand the 5' 3' polymerase activity of DNA polymerase 3, let's suppose that this is a DNA template. Now we already know that DNA polymerase 3 cannot initiate the synthesis of new strand, and it needs a primer to start the process. So first RNA primer reads the template strand in 3' 5' direction and adds nucleotides in 5' 3' direction, just a few. Now using the 3' OH of the primer, DNA polymerase 3 also reads the template strand in 3' to 5' direction and synthesizes a new strand in 5' to 3' direction by adding corresponding nucleotides to the 3' end of the growing chain. The new strand is complementary to the template strand, which means DNA polymerase 3 adds nucleotides following the base pair rules, that is, adenine pairs with thymine and guanine pairs with cytosine. So we can say that DNA polymerase synthesizes the new strand in 5' to 3' direction by adding nucleotides to the 3' end of the growing chain following the base pair rule. And this is what is called 5' 3' polymerase activity. DNA polymerase 3 exhibits 3' 5' exonuclease activity. So most DNA polymerases can check their work with each nucleotide that they add to the chain. So after adding a nucleotide through 5' 3' polymerase activity, enzyme DNA polymerase 3 comes back in 3' 5' direction and checks if it has added the correct base complementary to that present on the template strand. If yes, very well, it moves ahead and adds another nucleotide to the growing chain. Again, it comes back in 3' 5' direction to check if it is correctly matched to its complementary base on the template strand. Now, if it encounters that the base of the newly added nucleotide is mismatched to the base of the template strand, as you can see, the template base is A and polymerase has inserted G instead of T. So then enzyme DNA polymerase removes the incorrect nucleotide by making a cut at the phosphodiester bond via hydrolysis. This is called 3' 5' exonuclease activity or proofreading. After which DNA polymerase 3 exhibits 5' 3' activity and adds the correct base. Now let's see another example. As you can see, the template base is C and the enzyme inserted A instead of G into the new chain. Then the 3' 5' exonuclease activity of the enzyme will hydrolytically remove the misplaced nucleotide, after which DNA polymerase replaces it with G by exhibiting 5' 3' polymerase activity. So as each nucleotide is added to the chain, DNA polymerase 3 also checks to make certain that base to the newly added nucleotide is complementary of the base on the template strand. So what we can say is that if DNA polymerase 3 mispairs a nucleotide with the template, it uses its 3' 5' exonuclease activity to excise the mismatched nucleotide. As I told you earlier, this process is known as proofreading.
Next key feature of DNA polymerase 3 is that it synthesizes leading and lagging strand. To understand this, let's get back to one half of the replication bubble. So let's consider the lower strand where RNA primer is placed against 3' end of the template strand. Again, what would be the polarity of the primer? It will be anti-parallel to the template strand, right? So its 5' end will be here and 3' end will be here. Now using the 3' OH of RNA primer, DNA polymerase 3 synthesizes a new strand towards the replication fork continuously. This strand is called leading strand. So what is leading strand? Leading strand is the DNA strand that is synthesized continuously by the enzyme DNA polymerase 3 in 5' 3' direction towards the replication fork. Here you must remember that DNA polymerase 3 also exhibits 3' 5' exonuclease activity, that is proofreading or error detection, which we have already seen in the previous slide.
Now how about the other strand? From the center, the top strand is in 5' to 3' direction, and we have already discussed that no enzyme can synthesize DNA in 3' 5' direction. So now where should we place the RNA primer against the top strand? The primer will be placed at the angle of separation where the polarity of template strand is 3' to 5'. So reading the template strand in 3' 5' direction, primase synthesizes primer against the template strand. Now what will be the polarity of primer? It will be 5' to 3'. Now using the 3' OH of RNA primer, DNA polymerase 3 synthesizes a new DNA strand against the replication fork. Isn't it clear from the diagram that the new DNA strand is synthesized away from the replication fork and not in the direction of the replication fork, right? Now it is important to note here that DNA polymerase 3 can continue to synthesize new DNA strand until it comes across another RNA primer in its view. From where do you think this RNA primer would have appeared? I'm repeating again, from the angle of separation, DNA polymerase 3 synthesizes a DNA strand and it continues to synthesize the DNA strand only until it comes across an RNA primer in its view. From where do you think this RNA primer would have appeared? It then comes ahead and recognizes the 3' OH of primer and starts synthesizing a new DNA strand until it comes across another RNA primer in the view. And now you know where this primer came from, right? Another replication fork starts acting from behind and the same process continues. So against the top parental strand, you will be able to synthesize new strand only discontinuously. And so this new DNA strand is called as lagging strand. Yeah, what is it called? It's called lagging strand. So what is lagging strand? Lagging strand is the DNA strand that is synthesized discontinuously by the enzyme DNA polymerase 3 in 5' to 3' direction away from the replication fork. So as you can see, the lagging strand synthesis requires multiple RNA primers at specific intervals followed by synthesis of short stretches of DNA. These short stretches of DNA with RNA primers attached to them are called as Okazaki fragments. So what are Okazaki fragments? Okazaki fragments are short stretches of DNA with RNA primers attached to them. And they are being synthesized away from the fork towards the origin of replication, right? So here, along with the 5' 3' polymerase activity, DNA polymerase 3 also exhibits 3' 5' exonuclease activity simultaneously, that is proofreading or error detection. So if we compare the two strands, we will see that leading strand requires only one primer, whereas lagging strand requires multiple primers. DNA polymerase 3 synthesizes DNA in continuous manner on leading strand, whereas on lagging strand, DNA is synthesized in the form of Okazaki fragments. On leading strand, replication occurs towards the replication fork, and on lagging strand, replication occurs away from the replication fork. Now let's see the other half of the replication bubble. Here notice which way the replication forks opens? Towards this side, right? Now considering the polarity of the template strand in which the top strand is in 3' 5' direction towards the replication fork and the bottom strand is in 5' 3' direction towards the replication fork. So then which one would be the leading strand? So reading the top strand in 3' 5' direction, DNA polymerase will synthesize leading strand in 5' 3' direction, right? Similarly, reading the bottom strand in 3' 5' direction, DNA polymerase 3 synthesizes lagging strand in 5' 3' direction away from the replication fork, right? So the top strand will be the leading strand and the bottom strand will be the lagging strand. So now if we join the two halves, we will get the bubble, right? The replication bubble. And now you will be able to understand why replication is called bidirectional process and why within the replication bubble a strand in one direction will be leading strand and the same strand in other direction will be lagging strand, right? Again, coming back to one half of the replication bubble. So now we have the replication bubble like this. So do you think this is fair? One strand is continuous and the other is discontinuous? No, right? The Okazaki fragments must join to form a continuous strand and the RNA primer must be removed as well. So to solve this problem, another enzyme DNA polymerase 1 comes into play. So let's first see what DNA polymerase 1 is capable of. What are the key features of DNA polymerase 1 that allows it to remove the primers and solve the problem of Okazaki fragments on lagging strand. So the key features of DNA polymerase 1 are that it exhibits 5' 3' exonuclease activity, 5' 3' polymerase activity and 3' 5' exonuclease activity. So just like DNA polymerase 3, DNA polymerase 1 also exhibits 5' 3' polymerase and 3' 5' exonuclease activities. But in addition to these two, DNA polymerase 1 also exhibits 5' 3' exonuclease activity. So let us see what is 5' 3' exonuclease activity of DNA polymerase 1. So as you can see, the 5' 3' exonuclease activity of DNA polymerase 1 allows it to remove one or more nucleotides at a time from the 5' end to the 3' end of the RNA primer. So we can say that 5', 3' exonuclease activity of DNA polymerase 1 is nothing but removal of primer. Next is DNA polymerase 1 exhibits 5', 3' polymerase activity. Now as the primer is removed, DNA polymerase 1 has to fill the gap, so it adds one nucleotide at a time in 5' to 3' direction with the base corresponding to the base present on the template strand. So we can say to fill the gap caused by the removal of RNA primer, DNA polymerase 1 synthesizes the new strand in the 5', 3' direction by adding one nucleotide at a time following the base pair rule. DNA polymerase 1 also exhibits 3', 5' exonuclease activity. As I mentioned earlier, most DNA polymerases can check their work with each nucleotide that they add to the chain. So after 5', 3' polymerase activity, which we have just seen in the last slide, DNA polymerase 1 also comes back in 3', 5' direction and checks if it has added the correct base that is complementary to the base present on the template strand or no. If yes, very well, it moves ahead and performs 5', 3' polymerase activity and adds another nucleotide to the growing chain again. But if it encounters that the base of the newly added nucleotide is mismatched to the base of the template strand, then it removes the incorrect nucleotide by making a cut at the phosphodiester bond via hydrolysis. And what is the term I told you about 3', 5' exonuclease activity? It's proofreading. Again, after which DNA polymerase 1 performs 5', 3' polymerase activity and adds the correct base corresponding to the base present on the template strand. So we can say that if DNA polymerase 1 mispairs a nucleotide with the template, it uses its 3', 5' exonuclease activity to excise the mismatched nucleotide. And this process is known as proofreading. Again, coming back to the one half of the replication bubble, let's consider the lower strand, which is the leading strand. Now DNA polymerase 1 comes in and first removes the RNA primer exhibiting 5', 3' exonuclease activity, after which it fills the gap by performing 5', 3' polymerase activity and synthesizing new strand by adding nucleotides corresponding to the template strand. What else it does? It also performs 3', 5' exonuclease activity, which is proofreading, where it moves backwards in 3', 5' direction, and if finds incorrect base pair connection, it removes it, right? Now the same process happens in the lagging strand, that is 5', 3' exonuclease activity, 5', 3' polymerase activity, and 3', 5' exonuclease activity. But because of the Okazaki fragments on the lagging strand, even after DNA polymerase 1 fills the gap created by removal of the primers, still there is some space left. So to fix this problem, one more enzyme called ligase comes onto the lagging strand and fuse the DNA ends together so that the lagging strand becomes perfectly connected and continuous. So this was one half of the replication bubble. Now let's see what happens at the other half of the replication bubble. After that, we will add the enzyme polymerase and ligase to the list of enzymes and proteins. So even here in the other half of the replication bubble, after the action of DNA polymerase 1 and ligase, the lagging strand becomes perfectly connected and continuous. So if we join the two halves, finally our replication bubble would look like this, where we have elongated the DNA by copying the parental DNA and made the new daughter DNA. So now let's add the last two enzymes, polymerases and ligase, to the list of proteins and enzymes. So enzyme DNA polymerase 3 and DNA polymerase 1 synthesize DNA and also proofreads for error detection. And enzyme ligase seals the gaps between the two DNA strands on lagging strand. Now till here the stages of replication, that is initiation and elongation, are same in both prokaryotes and eukaryotes. But the only difference is the proteins that are used in eukaryotic replication. So before we head towards the termination stage, let's have a quick look at the different proteins and enzymes and their function in eukaryotic replication. So in eukaryotic replication, the origin of replication is identified by the protein ORC, that is origin of recognition complex. The activity of helicase in eukaryotic cell is done by protein MCM. The single-stranded DNA protection is provided by protein RPA. Primer synthesis is done by polymerase alpha or primase. A sliding clamp pCNA is used in eukaryotic DNA replication by enzyme DNA polymerases for the synthesis of daughter DNA strand, and finally the primer removal is done by RNase H and FEN1. Now, unlike the prokaryotic replication where we had seen only two DNA polymerases, type 3 and type 1, in eukaryotic replication there are numerous DNA polymerases perform different activities. Let's now check the activities of eukaryotic DNA polymerases as well. So polymerase alpha contains primase and it initiates the DNA synthesis. Polymerase beta performs the function of repair. Polymerase gamma replicates mitochondrial DNA. Polymerase delta elongates Okazaki fragments of the lagging strand, and polymerase epsilon elongates the leading strand. So here what you need to remember is that out of the 5 polymerases, only polymerase gamma, polymerase delta and polymerase epsilon performs the proofreading function. You must remember that polymerase alpha and polymerase beta do not perform the proofreading function during DNA replication in eukaryotic cells. So before we head towards the last stage of replication, that is termination, let's see a quick recap of elongation steps. First, you must remember that DNA polymerases can synthesize new DNA strand only in 5' to 3' direction. DNA polymerase cannot initiate the synthesis of new strand, it only elongates the strand by adding nucleotides to the 3' end of the RNA primer. And you must remember that RNA primer is synthesized by primase of primosome. Now, DNA polymerase 3 synthesizes leading strand in continuous manner in 5' to 3' direction by copying the 3' 5' strand of parental DNA. Similarly, DNA polymerase 3 synthesizes lagging strand in discontinuous manner in 5' to 3' direction by copying the 3' 5' strand of parental DNA. The short segment of DNA at the time of discontinuous synthesis at lagging strands are called Okazaki fragments. DNA polymerase 3 proofreads the newly synthesized DNA, removing mismatched nucleotides with its 3' 5' exonuclease activity. Leading strand requires only one primer, whereas lagging strand requires multiple primers. RNA primers are removed by DNA polymerase 1 by using 5' 3' exonuclease activity. DNA polymerase 1 fills the gaps with DNA by 5' 3' polymerase activity and proofreads the newly added nucleotides simultaneously by 3' 5' exonuclease activity. And finally, the nicks that are remained after the removal of the primers are sealed by enzyme DNA ligase. Finally, the last stage of replication, that is termination. First, we will discuss termination in prokaryotes. So this is prokaryotic circular DNA. This is parental strand. This is site of initiation of replication or IC. This is newly replicating daughter strand. Enzyme helicase on both the replication forks to unwind the parental DNA for replication to continue. And two replication forks moving in opposite direction. Now just opposite to ORIC, there is a site known as replication terminus. This is the site where DNA replication is terminated. So the replication terminus contains a series of sites called termination sites or TER sites. These TER sites are recognized and bound by TUS protein. So the TUS protein binds to this TER site and so a TER-TUS complex is formed. This TER-TUS complex acts as a trap for replication fork and cause termination. Let's see how it happens. So The TUS protein has one permissive phase and a non-permissive phase. The permissive phase allows the helicase to move forward, whereas the non-permissive phase blocks the passage of helicase for further movement. Now look at the helicase moving through the replication fork on the left. So as it reaches the TUS complex, it faces the non-permissive side of the TUS protein, right? And its further movement is blocked. Whereas the helicase that is moving through the replication fork on the right faces the permissive side of the TUS protein, correct? And thus it continues to move in, and finally both the helicases meet. This decreases the affinity of TUS protein for TUR site, and the TUS complex dissociates. Now, as the TUS protein dislodges, the polymerase moving along with helicase replicates the two strands, creating two complete daughter copies containing one parental and one new strand each. So let's recap the termination in prokaryotes. So at a certain specific points, the replication stops and starts terminating. That specific point is known as the termination site or TUR site. The TUS protein binds to this TUR site and forms TUR-TUS complex. TUR-TUS complex acts as a trap for replication fork by preventing helicase activity and further unwinding. This decreases the affinity of TUS protein for TUR site, and the TUS protein dissociates. As the TUS protein dislodges, the polymerase moving along with helicase replicates the two new strands. Thus, two complete daughter copies are formed containing one parental and one new strand each. Now, what about termination in eukaryotes? So, the termination in eukaryotes is different from prokaryotes because of the two differences in their genome. First, prokaryotic genome is smaller, eukaryotic genome is larger. Prokaryotic genome is closed and circular, whereas eukaryotic genome is open and linear. Now, because we eukaryotes have a larger genome, if we try to replicate all our 46 chromosomes with only one origin of replication, it is estimated that it will take 150 hours for all the chromosomes to be replicated. So that's why, to reduce the duration of replication from expected 150 hours to the actual 4 hours, we have multiple sites on our chromosomes which are 80 rich sequences. So multiple origin of replication is the classic feature of eukaryotic replication. So as replication in eukaryotes occur in several replication bubbles simultaneously, the termination involves merging of two adjacent replication forks. Let us see how. So at the two adjacent replication forks, the replisome, that is helicase and polymerase, gets dissociate, and the leading strand of one replication bubble eventually connects with the lagging strand of another bubble, and this is how termination occurs in eukaryotes. So this is all about DNA replication. Do check out the next video for N replication problem, role of telomere and enzyme telomerase in eukaryotic replication, as well as about replication inhibitors. Thanks for watching. If you found this video helpful, don't forget to give it a thumbs up, subscribe to our channel and hit the notification bell so that you never miss an update. Share it with your friends who might also find it useful, and stay tuned for more videos on other fascinating biochem topics. See you in the next one. Till then, happy learning.